This is the outline of my talk. I will first describe briefly which are the fundamental mechanisms of immunoregulation in the liver, how this applies to, or how this actually operates in experimental transplantation in animal models, and what are the clinical implications derived from this phenomenon in the setting of what we call operational tolerance in clinical liver transplantation. So everybody is well aware about the very important metabolic and detoxifying functions of the liver, and also that the liver is important by producing bile. But the immune-related functions of the liver are less well recognized. And indeed, the liver works as a lymphoid organ, but with a peculiarity in that immune responses taking place in the liver tend to be biased towards immunoregulation or tolerance. And there's probably a teleological mechanism why this is the case. Because there are a number of various neoantigens that are derived from this metabolic and detoxifying functions, and also because the liver receives a great deal of bacterial-derived products from the intestinal blood circulation. And there would be a risk for cytopathic immune responses being developed against these neoantigens and bacterial products unless this bias towards immunoregulation was operating. Now, to understand how this actually happens, it's important to remind the liver microanatomy. As you probably know, the liver receives most of its blood from the portal vein. This is a low-pressure system, and the liver, the blood through a network of very narrow vascular channels, the sinusoids, which are lined by a peculiar type of endothelial cells, which are fenestrated, which means that the lymphocytes circulating through the portal flow have ample opportunities to interact with the liver cells. Now, this is relevant because in the liver, unlike in other organs, there are many cells that are capable of presenting antigens to lymphocytes. So apart from the dendritic cells, hepatocytes, Kupffer cells, which are the largest compartment of tissue residing macrophages, sinusoidal cells and stellate cells, they have the capacity to present antigens to T cells. And they do so, again, in an unusual manner, because these cells, although they are capable of presenting antigen, they are actually not very strongly immunogenic, which means that when T cells recognize antigens presented by these cells, they tend to be activated, but to be subsequently deleted. In addition, CD4 cells, when they recognize antigens in the setting of in the liver, they are more prone to actually be converted to a regulatory phenotype. And this occurs almost universally under physiological steady-state non-inflammatory conditions, but in many cases under inflammatory conditions as well. Now, there are many mechanisms that are probably responsible to maintain this immature and low immunogenic profile of antigen-presenting cells within the liver. And the one that has been actually described in more detail is the LPS desensitization or tolerance phenomenon, through which the constant stimulus of LPS through TLR4, and this actually provides from the intestinal circulation, results in antigen-presenting cells being desensitized, and instead of actually producing pro-inflammatory cytokines, expressing a number of tolerogenic cytokines. And there are a number of other mechanisms that act in the same manner in at the level of the liver antigen-presenting cells. Now, I'm going to show just two examples of how powerful these tolerance phenomenon are. These are two relatively recent papers in murine models. So this is a paper published by the group of Patrick Bertolini in Sydney, where they transfer CD8 naive cells, TCR transgenic, into a recipient that ubiquitously expressed the cognate antigen for the TCR transgene. Now, in contrast to what one would have expected, no graft versus host disease occurs in this model, and the reason is because these naive CD8 cells They mostly accumulate in the liver, and they are very rapidly cleared from the liver by a combination of T-cell apoptosis and by a peculiar phenomenon that occurs in the hepatocytes, which is the hepatocytes actually engulf and phagocytosing the T-cells. And if these mechanisms actually inhibit, then these naive CD8 cells are actually capable of eliciting full-blown graft versus host disease.